Hi there. This is video 2, or the second part of phylogenetics, phylogenetic networks, and phylogenomics. And so in this one, I'll talk about some specific applications of phylogenetics that I've used in my own research. So this first one was to sort of sort out this fungal genus, genus Dermosobe, from another genus Cortinarius in the Cortinariaceae. So they're kind of a confusing set of fungi in that they can range anywhere from a purple color to a red color to orange and even green or olive caps. They're very difficult to identify and classify, so most times you need an expert to make sure that you have the right fungus in case you want to use it or consume it. So way back in, I think, 88, 89, somewhere in there, I suggested at a meeting to use the internal transcribe spacers of the ribosomal gene locus in a ribosomal RNA gene locus in the nuclei of fungi. So it was way back then we first suggested that. Now it's pretty popular to use this region. And the reason is, and I talked about this last video, I believe, that the small subunit and large subunit are way too conserved, highly conserved. So you can't tell one fungus from another, really, from that area. And even within the spacer region, the 5.8S gene is too well conserved. But if you move out into these internal transcribe spacer regions, they have some conserved regions and some variable and hypervariable regions that can be used to distinguish one species from another, and they can be used then for phylogenetic analysis to classify members of these taxa. And by the way, this ITS region is useful for many types of fungi. So you could even use it in a clinical setting to identify a species of fungus. So this outlines some of this genus up here with all the Ds. Those are Dermosobes. Then the Cs down here stand for a closely related genus Cortinarius, and you can see some Ds in here. These are those olive-colored ones that are within the Cortinarius clade, so that was a problem right from the beginning. The samples that we used had been collected between 1927 and 1985, so these were dried and preserved specimens, including what's called the holotype of Cortinarius limonius. And so this holotype is the specimen that was used to first describe this genus and this species back in 1927. So we were trying to get as close to the original definitions for these genera as possible. So after extracting the DNA and then amplifying the ITS regions by PCR and sequencing them, then we went through phylogenetic analysis. And in this case, we were using maximum parsimony, which I described briefly in the previous video. So from this, you can see the historical classifications, including Dermosobe up here, the genus Dermosobe, and then sections. So that's a sub-taxonomic group. So right away, we could, from this phylogenetic analysis, we could see that there were some problems. So here are those sections. Section Dermosobe, that looks pretty good. Section Sanguinea, part of it's here. And then part of it's up here split by section Malacoriae. So that means these are paraphyletic, so that they're not in natural groupings. So we suggested that now there's a section Dermosobe, there's a section Sanguinea characterized by Dermosobe sanguinea, and then there's a separate section of Sanguinea that's primarily identified by Dermosobe semisanguinea, a slightly different species. And so this entire part up here is sanguinea and dermosity, and we get rid of this one called Malacori. It doesn't seem to be a natural taxonomic group. And then down here within the Cortinarius, the genus Cortinarius group, there were several different subgenera that had been proposed previously. And so we accepted some of those, and others of them seem to be lacking in definition. And then there are these two here. These are within subgenus Dermosity. And so these ones, we're not quite sure where they should go, but if they truly are Dermosity, then they're not coming out in the right place, and they look more like a Cortinarius species that could be a subgenus of Cortinarius. So still a few problems left over even after we did our phylogenetic analysis. So this was published back in, 89 or 90. And it's been cited several times as being, the definitive characterization of Dermosity. Now, on this phylogram, here's a close-up. You can see there are numbers on some of those branches, and those are bootstrap values, and that's a way to statistically test the strength of each one of those branches, how much they're supported by the data. So bootstrapping is known as a resampling method with replacement. So if we have this character matrix here with the OTUs over here and then the nucleotide positions here for comparison, then the program or the algorithm will calculate a tree based on that, and that'll be the tree output there. So that represents tree number 1. Then the computer will go back and start resampling randomly. So it might take character 9 first and put that in this position of the matrix. And then it'll go back and randomly take another character, in this case character 2, and place that in the matrix. And then it'll go back and select randomly another character. In this case, it's number 9 again. And that's where this replacement comes in. So even though it's selected 9 once, it can select 9 again because it's replaced it in that original matrix. Then it selects another one, in this case character 5, and eventually builds up a matrix that's equal in size to the first matrix and creates a tree based on that data matrix. So it's slightly different. Then it does the resampling with replacement on a third matrix, makes a third tree, and does this over and over and over many times, usually 500 to 1,000 times. 1,000 is sort of the norm. And it then calculates the frequencies of how often particular branches appear on each one of those trees. So if a branch appears in 1,000 out of 1,000 iterations, then that is strongly supported, and that gets a value of 100 or 100%. If a particular branch gets only 40, is there only 40 times on those trees and is missing from the other 60%, then that means that it's only supported by 40%, and usually the cutoff is 50%. So if it's 50% or higher, it's counted as being significant. So again, here's a close-up of that. So this top one is supported 99% of the time, 98%, 75%. So greater than 70% is considered good support, and less than 50% are not shown. So on some of these branches, those branches are not supported by more than 50% of the trees that were generated from this bootstrap analysis. A more statistically robust method of phylogenetic analysis is maximum likelihood, but it really takes a lot of computing time. So in some cases, its use is limited, but in some cases, it can be used to test hypotheses. So in that, it is good in certain situations. But essentially what maximum likelihood does is it takes a sequence on one branch. So it builds the tree first, just like neighbor joining a nose did. And it joins the closest two together, and then the next one out, and the next one out, builds this tree. And then it calculates on each branch what the probability of the beginning sequence is to change into the ending sequence along that branch. And that's based on the models that you provide, whether you have transitions or transversions of equal value or unequal value. And it also looks at the amount of data in the data set, how many A's, G's, C's, and T's are there, and then calculates a likelihood for the change along each branch. So here you have the ln likelihood of that branch there, branch A, branch B, and the tree then is the sum of those ln likelihoods that were calculated for each branch. So we used it to test out a few things. So we were testing hypotheses using maximum likelihood for those Dermaceae species. So you can test it for things like, is taxon A, should it be on the same branch as B, such as humans and gorillas? And we were looking for different taxa of Dermaceae and Cortinarius. Also, you can compare it with the maximum parsimony tree. Is that congruent with the maximum likelihood tree? And you can also look at it as far as gene families. Is gene A in the same gene family as B? So a lot of different ways to use maximum likelihood apart from just determining an overall tree. So going further with Dermaceae then, we see, it ranges all the way from this, which that's actually a Cortinarius, this purple color, and then the reddish and orange and yellow colors in Dermaceae, and this is Olivacea picta, which is an olive-colored one. That's the one that kind of pops into the clade that includes Cortinarius. So, where do these actually go? And you do this by then comparing the maximum likelihood tree with these other trees. So there's a way for these programs, you enter the maximum likelihood tree, and then you can actually enter into the algorithm or into the program the maximum parsimony tree, which you see have a few differences in the tree lengths, and then ask whether those are different. So in this case, they were a few differences in steps. This is the ln likelihood of the maximum likelihood tree, the ln likelihood of the maximum parsimony tree, then the standard deviation between those. This one is the best tree, and the maximum parsimony tree, which was almost the same, was not rejected, meaning that both trees are equally likely given the data that we were using. So then we fed it another tree which was constrained. So we put all of the Dermaceae together in the upper part of the tree, and it also was not rejected. We had another tree that was constrained, so we pushed that Dermaceae Olivacea picta within the Dermaceae, and it actually was rejected. So it's clear that that one Dermaceae that fit into the Cortinarius part of the tree cannot be pushed up into the genus Dermaceae, so it probably should be renamed a Cortinarius species. And we did two other groupings based on some previous papers, how they had placed some of these, and both of those also were rejected by this maximum likelihood test, meaning that the final tree that we found and we reported was most likely an accurate tree and depiction of both the genus Dermaceae and of the members of Cortinarius that we analyzed. So the last method that I want to mention is Bayesian phylogenetic analysis, and it has some similarities to maximum likelihood, but it takes less time, and it's an iterative approach so that the algorithm actually pops into a certain place in joining the taxa together, and then goes stepwise back and forward and backward by changing some of the branches to try and hone in or close in on the most likely tree. So, in words, what you're trying to measure is what the tree that you're looking at right now looks like, the prior tree, either one chosen randomly or you can select one, and then calculate from the data the likelihood of that tree changing to another tree that it's being compared to at the time. If it's more likely that the posterior tree, the one that's being examined, is better, then that one's chosen, and the calculation is done again with that one being the prior one, and then looking at the posterior, the next one. So it works its way through, and there's actually what's called a burn-in time, where it's sort of doing these large movements throughout the data and throughout the possible trees. And then after that, and that can be a million trees that it searches through. And then it's honing in on the best possible, the most likely tree, statistically speaking, finally coming to a consensus in the end of a tree that is likely the best one based on the data. So this is a little bit more about the calculation. So the posterior distribution is the forward-looking one, is based on the prior distribution times the likelihood of the change, and then a normalizing constant. And so this is it. The posterior distribution is based on the tree, the parameters, and the data. And then over here you have also a tree and parameters of the prior distribution, or prior tree, and then the parameters over here over the total data normalizing constant. And I haven't used this one very much, so I can't tell you a great deal, but there are videos on YouTube that you can find that go into a more thorough explanation of Bayesian phylogenetic analysis. And this is that same equation that then summarizes the theta parameters as the tree topology and the branch links over here. So now I want to talk a little bit more about some of the applications of phylogenetics that I've used in my own research, going a little further. And one of them was try to answer a question that has really bothered me for a long time. What's the origin of gram-negative bacteria, which have two membranes? Where did those two membranes come from? Because to have two membranes, and they're somewhat different, and the proteins are different, that requires a lot of genes to construct each one of those membranes and embed those proteins in there. So my major question was, did those two membranes come from two different organisms? And was there an endosymbiotic event between two gram-positive bacteria that led to the origination of the gram-negative bacteria? So several years ago, a study was done looking at specific genes in both monoderms and diderms to try to answer this question. And the problem is here. So if you look at a general phylogram of bacteria, you find that the monoderms, signified by the ones in parentheses here, are interdigitated between the diderms, the ones with two membranes, which means they're polyphyletic. That is, that the members that have one membrane versus two membrane are sort of interdigitated or scattered among one another. However, if you propose that there was an endosymbiotic event between two monoderms to form the diderms, and this was done by Lake from UCLA a few years ago, you find that you can have an endosymbiotic event joining these two together, and then the diderms become monophyletic. Now, it seems like, okay, everything was solved, except several papers came out after that criticizing it and saying it can't be because some of the statistics used were not reliable, some of the genes used were ones that shouldn't have been used, and other genes should have been used, so then it would come out very differently. But that kind of intrigued me that you could have two monoderms coming together to produce the diderms. So if you look at cell wall and cell membrane structure, you find the monoderms up here. Thermotoga is an oddball, and then you have some atypical diderms over here, negativicutes, and others, Fusobacterium. And then Corynebacterium up here that has a mycolic acid layer in its cell wall. Then you have Chloroflexi, which is a monoderm also, but has a sort of uncharacterized outer layer. And then you come down here into these diderms that have lipopolysaccharides in their outer cell membrane, which is characteristic, although these ones also have that lipopolysaccharide in their layer. So you can already see that there's somewhat of a complex picture between the monoderms and the diderms. So in addition to the cell membrane and cell wall differences, there are some sequence differences in ribosomal proteins, in elongation factor proteins, in pyruvate kinase, etc. So you can also use phylogenetic analysis to make these points of branching into the various monoderms and diderms. But what originally got me more interested in this is that if you look at the monoderms, and those are right up in this group here, the monoderms are all together, and the diderms are over here, and so those are most of the diderms except cyanobacteria. So cyanobacteria is over here, and cyanobacteria also are unique in that they have both photosystem 1 and photosystem 2, or reaction centers 1 and reaction centers 2, while the other monoderms and diderms have either reaction center 1 or reaction center 2, but not both. So my focus then was on photosynthetic apparatus as well as cell membrane and cell wall components. So another key to this was not only to look at the photosynthetic apparatus, but also to look at the bacterial chlorophylls. And so this is some trees not done by me, but done by others on bacterial chlorophyll F. And it sort of joins these things up into the different groups. Here are the diderms up here, primarily proteobacteria, but then Chloroflexi in here is a monoderm, and then you go to this other part of the clade over here, and that's Chlorobi, which is a diderm, but having a different type of reaction center. And this is sort of a summary of it, and it's kind of blurry, but that's the best picture I could get. But this orange one here, that's cyanobacterium, so you keep your eye on that one. And then Chloroflexi is a monoderm, and Actinobacterium also is a monoderm. And if you look at the various photosystems in bacterial chlorophyll F, you can see that Chloroflexi here in purple is closer to the acidobacteria and proteobacteria here than it is to Chlorobi, so it's further away. Whereas for reaction center 1, Chlorobi and acidobacteria are very close together, while for reaction center type 2, proteobacteria and Chloroflexi, which are a diderm and monoderm respectively, are very close together. And all the time in this sort of background tree, this phylogenetic tree in the background, you can see that cyanobacteria is fairly close to Chloroflexi, a monoderm, as well as the Firmicutes, which are also monoderms. And you might say Chloroflexi, yeah, that's photosynthetic, but you might say that Firmicutes are not photosynthetic. But There are members of the Firmicutes that are photosynthetic, and they have reaction center type 1, and chloroflexi has reaction center type 2, and both of those are found in cyanobacteria. So just to show you what's involved in those reaction centers, here's this ancestral type. It then diverges very early in evolution, splitting into a type 1 reaction center and a type 2 reaction center. These go off and form chloroflexi and proteobacteria, so those are in there. And then the type 1s are in acidobacteria, chlorobi, and heliobacteriaceae, which is in the Firmicutes. And then this crossover here is due to the fact that cyanobacteria have both reaction center 1 and reaction center 2. But while modern cyanobacteria, starting around 2.7 to 3 billion years ago, are oxygenic because they picked up a manganese center to help with that process, these early ones back here were anoxygenic. That is, they did not produce oxygen. Still did photosynthesis, just did not produce oxygen. And these ones over here do not produce oxygen either. So in building up this story also, I had to look at the biosynthetic pathways for producing each of the chlorophylls, including the bacteria chlorophylls and then eventually the chlorophylls in the cyanobacteria. And so I considered those also to map onto a phylogenetic tree. So I started with just this part of this tree, which is all the bacteria, all the known bacteria, and began to rearrange some of the branches on that tree to reflect the type of photosynthesis they had, the reaction center they had, the type of bacterial chlorophyll they had, and also those genetic changes in the ribosomal RNA, ribosomal protein genes, the elongation factors, etc., to try and come out with something that actually made sense from a molecular standpoint. So if we start down here with this ancestral reaction center in the bacteria, the archaea, and eventually the eukaryotes go off in that direction. Now we have a split between these three groups of monoderms, the Firmicutes, which one group has reaction center 1, which diverged from reaction center 2, which appear in the chloroflexi monoderms. And then actinobacteria aren't known to have any photosynthetic members. So you can also see the bacterial chlorophyll are evolving at the same time, or more are being added. So start with bacterial chlorophyll A, moving into bacterial chlorophyll G, and up here A evolving into bacterial chlorophyll C, and just minor changes in each one of those, but then they have different absorption maxima. So by creating an endosymbiotic event between a chloroflexi and this member of the Firmicutes, then you can create an organism with both photosystems, and those would be the cyanobacteria. To move into the other species, then they would have to lose either one or two of those, one or both of those photosystems, and loss of those, which entails many, many genes, would be fairly simple just by mutating one gene or two genes at a time, and then you can very easily, without selection anyway, lose that ability to be a photoautotroph. This is also consistent with those changes in those genes. So I've listed those gene changes as 1, 2, 3, 4 through 12, 12 different mutations that have been recorded. So 1, 2, 3 occur and 4 and 5 occur here within the monoderms already. So they're present in almost everything except the Firmicutes and actinobacteria. Remember I said the placement of Thermotoga and related taxa was difficult, and I think the reason is it comes out way down here. It's a very ancient taxon, and it only has 2 of those mutations and is missing mutations 3 through 12. So its best place to branch out is right here, all by itself. And then a couple more mutations in chloroflexi, and then you can see one by one along these lines. The number 6 mutation appears there, first 7, 8, 9, 10, and 11, and 12. So all the way out here to the end in the gamma proteobacteria, they have all 12, whereas the beta proteobacteria have mutations 1 through 11, alphas have 1 through 10, etc. So now this is consistent with photosystem type, those mutations, as well as the bacterial chlorophyll type. So we've got, fairly good consistency then, joining a chloroflexus together with a heliobacteria in the Firmicutes to form all of these diderms over here. But these ones were always a problem. So Deinococcus, Thermus, Fusobacteria, Negativicutes, and similar groups. And those can be explained then because they don't have any of these mutations over here that you see. That is, they're missing mutations 2 through 12. So they don't have any of that. So that's the first clue that they don't go with this other group. And Negativicutes have been classified as monoderms or diderms, and so they're kind of all over the place. But they can be explained by a second endosymbiotic event between an actinobacterium and a Firmicutes to form those, and then that's consistent. That makes this entire tree consistent with all of the characteristics that are found in those different organisms. So in this case, I've taken a previously published phylogram based on hundreds of species, and mapped on that photosynthetic apparatus and some other mutations that weren't included in the original analysis to try to elucidate where the diderms originated. And it looks like 2 separate groups of diderms originated from a couple of endosymbiotic events between 2 monoderms. And this is sort of a simpler diagram just showing the photosystems and how these 2 events led to modern bacteria. So you have this ancestral reaction center that evolves into 2 different reaction centers that have different centers in them, different ways of transferring electrons. Here's a reaction center down here, which is the Heliobacteriaceae in Firmicutes, and then the Chloroflexi reaction center type 2. So this main endosymbiotic event in black joins those 2 together, and you get the cyanobacteria that are at first anoxygenic and then eventually oxygenic. And then you have these other groups of diderms, including Lucimicrobia, which in the PVC group, which have no photosynthesis, so they've lost those reaction centers. Here's Gematomonadetes, which has reaction center 2, so it's lost reaction center 1. These 2 Chlorobian acidobacteria have lost reaction center 2. And then going into the proteobacteria, some of which are photosynthetic but have only reaction center 2, and others that have lost both reaction centers. But then here is the other proposed endosymbiotic event between an actinobacteria and a Heliobacteriaceae, or just a Firmicutes without photosynthesis, because these ones aren't photosynthetic anyway. So that's Deinococcus, Thermus, Fusobacteria, Halanorubiales, and then Negativicutes, which are also non-photosynthetic. But remember, if you look at the number of mutations here, they don't have the mutations of those genes that some of the others have. So the only way that they can be explained is by a second event that must have occurred. And incidentally, that's similar to the one that was proposed in that other paper that I alluded to earlier. A second project that I did recently was looking at evolution of the genetic code. So how did a complex code like this begin and evolve? So if you look at the genetic code this way, there's the first position, second position, third position, and all the amino acids encoded by those triplet codes. Everything in orange here, those are nonpolar amino acids. The purple ones are polar. The blue ones are basic, that is, they have a positive charge. And the green ones are acidic, aspartate and glutamate. And then there are 3 stop codons. And this is, quote, the universal genetic code, but there are differences in the genetic code in mitochondria and then some protists and others. So it's not quite universal, but it's mainly universal. Nonetheless, this separation between nonpolar, polar, etc., indicates there's some structure in this genetic code that must reflect something about its evolution. So here are the categories of some of the functions. You have this green group down here, the nonpolar ones going from alanine and valine, which are rather small, all the way up to tyrosine and tryptophan, which are much more complex. And then here are the polar amino acids, including serine, threonine, etc. And these 2 groups together form alpha helices and beta sheets. So those are important for those regions, as well as spacing out different parts of the protein molecule. And then these ones up here are the amino acids that are charged, including the positively charged or basic amino acids, arginine, histidine, and lysine, and then the negatively charged ones, aspartic acid or aspartate, and glutamic acid or glutamate at pH 7 anyway. And then you have these alpha helix breakers, which include glycine, which allows free rotation around the polypeptide chain. Cysteines, which can form disulfide bridges between distant parts of the molecule. Proline, which forms kinks, and then selenocysteine, which is used in some proteins. So early on in evolution, that is before any biology existed on Earth, 4.2 to 4.5 billion years ago, these amino acids were present either on Earth or they were being brought to Earth on meteorites, which carry these same amino acids. So all of these amino acids can be formed by non-biological means and have been found to do so, to be present in pre-biological conditions on Earth. And by the way, these 5 here are on the bottom row of the genetic code. So not only do you have some structure between nonpolar and polar and things like that in the genetic code, but the bottom row gives another hint that these may have been the starting point for the genetic code on Earth. So there they are, valine, alanine, aspartate, glutamate, and glycine. And you note that their structures are very simple too. So these are some of the simplest amino acids, including glycine, which is just a hydrogen, and alanine, which is a methyl group. And you see that they all start here from a nucleotide, the G nucleotide, which also hints at maybe the original genetic code early in evolution. Then if you go back to this chart, then these ones in red are all those ones on the bottom row. So you have one here in these charged amino acids, one of these which allows free rotation, and then something that's capable of forming alpha and beta sheets. So you have sort of one from each group in that original system. The ones in black are the ones that aren't on the bottom row, but also were very early, present very early on Earth. So this means even before there was any biological organism on Earth, there were many of the little pieces present, the molecules present, both amino acids and nucleic acids, by the way, primarily ribonucleic acid components, which were present before any organism existed on Earth. So then, building up the story a little further, if you look at the frequencies of amino acids in the most ancient proteins, and the most ancient proteins are those that are in every organism on Earth. That is, if they're in every organism on Earth, they must have existed very early, because in very modern proteins, they're limited to certain types of organisms. So here you see they are alanine, valine, glycine, and glutamate, and then over here is aspartate. So you have those 5 from the bottom row are in the highest frequencies in ancient proteins, also sort of confirming that they were present early on in evolution of the genetic code. And not only do you have those ones on the bottom row of the genetic code, but these other ones circled in black, those are the 10 amino acids that were present very early on Earth, including proline, serine, threonine, lysine, etc. So when you try to reconstruct the evolution of the genetic code, you have to take these things into consideration. If you look in modern proteins, then, you see that alanine, valine, etc., that are shown in red down here, are decreasing in frequency, whereas these ones in green, which are more complex amino acids, have been increasing in frequency, meaning that these ones were later arrivals into proteins and into the genetic code. So that helps to then start formulating some direction and system for recreating the evolution of the genetic code. Another set of evidence for the evolution of the genetic code comes from looking at these aminoacyl tRNA synthetases. These are the proteins that attach the amino acids to the specific tRNAs. So now you see a little more structure. Here's the class 1s in yellow and the class 2s in blue. You can see this lower row down here has an equal number of class 1s and class 2s, and those again are the earliest additions to the genetic code. And then the next things that happened were that the genetic code actually moved up on the left-hand side of that diagram. Phylogenetic studies have also been completed and published on how these aminoacyl tRNA synthetases, class 2 in this case, attach to the tRNAs and where they attach to the tRNA. So you can see here is attached to the acceptor arm and only barely touches the anticodon later in evolution. And here are the class 1s. You'll notice that the tRNA is facing the opposite direction because they attach to the opposite side of the tRNA. And in this case, it takes a long time in evolution before those actually touch the anticodon. But they recognize the acceptor arm here as well as other parts of the tRNA before they recognize the anticodon. So the anticodon wasn't the initially important portion of the tRNA for attaching the specific amino acid to that tRNA. So this just summarizes the evolution of those two types of AARSs. Here's class 1 and class 2, and the class 1s attach on one side of the tRNA. The class 2s attach to the opposite side of the tRNA. And you'll notice that they very early on, they don't even attach to the anticodon, but they're very specific for the acceptor arm where they're attaching the amino acids. So here's an overall evolution of those AARSs, as well as the evolution of the ribosomal RNAs, both the small subunit, large subunit, and the tRNAs. And you'll note the mRNAs in here are double-stranded for a time, and that has something to do with how these amino acyl tRNA synthetases attach and how the genetic code originally might have been read off in one direction by one set of amino acyl tRNA synthetases to add the amino acids, and in the opposite direction. So you have two different things happening at the same time: charging of the tRNAs with the amino acids, as well as translation that's occurring in close relationship to those. So again, you can see down here when this is arranged by class 1 and class 2 AARS, there's sort of an alternating pattern, meaning you can read off a type class 1 AARS here and a type 2 AARS here, one of each type here, and then type 2 over here. And going up, they seem to be matched too in a certain logical pattern. And if you assume that these are the initial amino acids here, then the next ones to appear are those other 5 that were available early on Earth, and so you can move up this direction on the genetic code table. Also, if you organize this according to whether the amino acyl tRNA synthetase attaches to the minor groove site versus the major groove site of the tRNA, you can see that that same pattern is there, and it's even further strengthened or further confirmed in this table as well. So again, this indicates that you have these this first group down here of 5 amino acids, and then going up in this direction to add another 5 or 6 amino acids to the genetic code. Then if you start examining these in a different way, to split them out a little bit, you find that you have self-complementary RNAs in this case. So for these ones, you read YUN, and in the opposite direction, it's NAR to get these amino acids, and you can find those on the genetic code table, and all of these here are, well, most of them are a class 1, which approach from the minor groove, and then the black ones are primarily class 2 that approach from the major groove, although there are a few exceptions in here as well for tyrosine and lysine and a few others. But the exceptions in here are some of the later arrivals to the genetic code. So from all of that information, then I built up a parsimonious sequence of steps that would lead to the current genetic code, and that's starting with these ones, which are in this sort of shaded L-shaped piece here. Those are those 5 on the bottom row. So you start there, and it looks like you start from alanine. And the reason I started from alanine is I went from the frequencies of alanine within the ancient proteins. So these are arranged according to the fact that they're the first ones, but they also are among the highest concentrations or frequencies in ancient proteins. Also, the steps are in 1 nucleotide differences. So going from alanine to glycine, you go from a GCN to a GGN. That is, only that middle nucleotide has changed, and it's a transversion. Going from alanine to aspartate, then you go from GCN to GAY, and again, that's a transversion of that middle nucleotide. Now, going from alanine to valine, now it's read in the opposite on the opposite side by that amino acyl tRNA synthetase. So, according to that last table that I showed, then you have to use the reverse complement of the CGN to create this one, which would be GUN. So you're really changing a C to an A on this strand, but it's a G to an U change over here. And so that's how I went about assigning these. So I've essentially used a maximum parsimony method to go in single nucleotide steps, adding the amino acids one by one. In doing so, then you see the ones with the double lines around them. Those are the those 10 that were on Earth very early, and you can see they're all clustered in this area of this diagram. The ones that have only single lines around them arrived a little later, and the ones with dashed lines are the ones that appeared later and are in the lowest frequency in ancient proteins. I've also superimposed on this, you see the little P's and O and F. Amino acids can be formed off of the TCA cycle as well as other closely allied biosynthetic pathways. So these ones are close to pyruvate. This is oxaloacetate and fumarate, etc. And you can see where these could be formed very early in evolution from that TCA cycle, which is a very ancient metabolic pathway. They can also go in reverse, so the amino acids can be used, alanine can be used to form pyruvate, etc. So those are reversible reactions. In addition, you can see that the molecular weights in parentheses here generally increase as you go further down on this chart, indicating the progression to more complex amino acids. These two groups, this one over here, threonine, serine, and proline, and then this other group, valine, leucine, isoleucine, and methionine. Each of those are added to homologous tRNAs. That is, those tRNAs are very, very similar to one another and are members of the same, genetic line essentially. So those go together also. Those, those were added afterwards just to show some strength in this. This evolutionary phylogeny that I've built. So there you go. There's just a few ways, and we've used phylogenetic trees and other ways to indicate what a species is, especially from our Antarctic work, pulling organisms out of the ancient ice to find out what they are. So you can use phylogenetics to do that as well. But these are just a few examples of some of the things that you can use with phylogenetic and phylogenetic type studies and methods. In addition to phylogenetic analyses, there are also phylogenetic network analyses. And these, with these, you can graphically represent unresolved branches. You can indicate potential horizontal transfer events. Also, they can indicate potential hybridization events and also potential recombination events. You can determine the resolution of the data sets this way and also study some endosymbiotic events and study multi-gene evolution. So these are a little more complex to deal with, but they can be very informative of some of these different ways that genes are inherited and evolving in different lines of organisms. So if a phylogenetic analysis yields these diagrams on the left, they yield straight lines between those, but then often the resolution is a bit odd in some of these. So you'll look at some of these bifurcating trees and find out that there are multiple ways of getting from A to C or A to E. And if the data are then analyzed in a different way, you can find that these are networks, so that there are multiple ways of going from, for instance, A to C and A to E. So instead of just having nodes and branches on the left, then there are branches, but there are also splits and edges. So the splits are these intersection points, and the edges are these edges of these polygons. So here's one example of a phylogenetic tree on the left, and it shows these nice bifurcating trees. But if then you reconsider the data and where these bifurcations are, and how many steps it takes to go from B to C and B to A, etc., then you find that a network can be formed based on that same data, so that going from A to C and A to F, etc., there are multiple pathways to that are either equally likely or at least close to one another in their possibilities. Some of these can be caused by recombination events in those organisms along certain lines, especially frequent recombination events. So you can get deletion and duplication events. Then there are intragenic exchanges, interchange, translocations, and transposition. And these can all affect what the possible lines of evolution and inheritance are along these. And so often these will lead to various combinations of alleles within each one of the lines of evolution, and allow for more than one possible track for each one of going from one point to another. So here's one phylogenetic network of blue tongue viruses, and it probably is happening similarly with the current coronavirus, where you have, instead of a single path leading to each one of these variants, there are multiple paths, often because of evolution within an individual, where they may be infected by more than one organism, one virus, or the virus is actively evolving in them. And so it takes several generations in a host to then come out with individual speciation or variant lines. And so this is more of a real representation of what's happening during these speciations and emergences of these variants, in that there are several things going on at the same time. You can sometimes get straight lines coming out of that when there's a very distinct speciation or variant evolution. But often there are interchanges, recombination, horizontal gene transfers, etc., that are going on within each one of those lines, eventually turning out to be an individual variant or species. Here's another tree looking at a broader range of organisms, bacteria and archaea. And so you can see in the middle here all of this network of paths that led to the emergence of each one of these groups, including the alpha proteobacteria here, the gamma proteobacteria, beta proteobacteria over here, and then the emergence over here of the archaea. And eventually you'd see one of these lines emerging into the eukaryotes as well. But this is more indicative of what's happening in evolution, that there are things being transferred back and forth. There are hybridization events and many other possibilities for inheritance of certain genes and characters. So such that each one of these lines that you see up here, these inferred evolutionary phylogenetic trees, if you really look in what's happening evolutionarily, it's more like this lower diagram where they aren't straight lines, but they're convoluted lines consisting of genetic interactions among the individuals in the population, as well as interactions with species around them. And so you have things like bottlenecks where the genetic diversity is lowered because of disease or famine or something else, and then you emerge later to form a larger population. And this actually happened with Homo sapiens, our species, where there was a bottleneck somewhere around 200,000 years ago. Emerging from that was, most of the genetic diversity diverged since then, but much of the genetic makeup of our species was concentrated right at one point at that time. So we almost didn't make it out of that bottleneck. And here's one line where there is an extinction event where the organisms don't survive. There are times when populations diverge and then they come back together, as well as times when two lines that have speciated into two nearly separated species come together in a hybridization event. And this has occurred in the genus Canis, mainly Canis lupus, which includes wolves and dogs and coyotes, both red wolves and gray wolves, and some jackals. In fact, the things that people call coyotes around here are actually coy wolves. They're a hybrid between wolves, gray wolves, which are primarily living in forests and have pack activity, and coyotes, which are primarily out on the prairies and are mainly singular animals. And the coy wolves are intermediate between that. They seem to have mated. There was a mating between a gray wolf and a coyote in the northeast U.S. or southeast Canada because of the deforestation caused by people in those areas for farming. That then pushed the wolves further to the northeast and allowed the coyotes to move in from the west. And there was a hybridization event somewhere in the 19 teens, somewhere in there, that created this new hybrid species called coy wolves. Their morphological characters are in between, so they're larger than coyotes and smaller than wolves. Their snouts are also different, intermediate between, and their behaviors are in between. So the coyotes normally hunt individually, the wolves hunt in packs, and the coy wolves can hunt in packs or individually. They've been seen to do both. And if a phylogenetic analysis is done on those, it would indicate something like this, where you have characters from both of these coming together into a new species or hybrid. So that's indicated here, where you have an individual line of organisms, the genus here, and then you have speciation into two separate species, wolves and coyotes, for instance. Those come together to form a new species. So now you have something intermediate between the two, and you can see that phylogenetically. So instead of forming a nice bifurcating tree, you get this circular pattern within the phylogram that you can clearly see, which is a joining of the two. And this happened with mitochondria entering eukaryotes. It's happened in human populations too, where you and I are hybrids of Homo sapiens and Homo neanderthalensis, and probably Homo denisova also, and maybe Homo erectus also. There have been hybridization events in the past. Additionally, horizontal gene transfer of sequences from one species into a different species, or into the same species occasionally, but mainly into different species, has occurred very frequently in the past. So you have two separate species here, and then you get a transfer of some genes from one species into another, creating a new sort of modified species B over here. So one study showing that included 181 genomes of bacteria. This diagram here is a phylogenetic tree based on ribosomal RNA genes. So it's a nice bifurcating tree that you see here with the species here up at the top representing the sequences that were obtained in these 181 genomes and then rebuilding back in the past to create this tree. However, if you look at certain sequences that do not fit with the ribosomal RNA gene pattern, but instead have been transferred from one species to another, and especially ancient lateral transfers, in this case, these green lines, indicating genes that have originated back here and then been moved along these branches and across these branches into other species that had separated sometime before or after. You can also isolate genes and sequences that are more recent transfers, not very recent, but more recent transfers. And you can tell this by the number of changes that have occurred since they were transferred from one species into another. So there's more lateral or horizontal gene transfers that have been occurring more recently. And then this diagram is the gene transfers, horizontal gene transfers that are the most recent, only occurring very recently, and you can tell that because they have not changed much at all since their transfer. So then if you superimpose all of those horizontal gene or lateral gene transfers, you get a pattern like this, which is sort of like a spider web, right? So you have the overall ribosomal pattern where things have been inherited vertically, but then all of these other sequences have been transferred, some a long time ago, some more recently, and some very recently. So they're occurring all the time, and they have been essentially since the beginning of life on Earth. So this also is an indication that evolution isn't a simple bifurcating mechanism, but things are being transferred all the time, which then makes the phylogenetic networks make more sense, that things are being tossed around from one line of the phylogram to another line of the phylogram. Another aspect of phylogenetic analyses is that it's shown us sort of new horizons in a way. This phylogram is of hundreds of different species. And so on the left over here, this left half, essentially, those are the bacteria. The monoderms or gram-positives are up here, the gram-negatives are down here, except cyanobacterium. And then the archaea are down here. And this one line of archaea, those are the eukaryotes. So you can see that the eukaryotes, including us, originated from one line of archaea, and there's not a great deal of separation between them. But we are also, the archaea picked up a alpha proteobacterium that became the mitochondrion, and a cyanobacterium that became the chloroplast in the plants. And there were other endosymbiotic events too. But this branch that's in purple over here with the red dots, it says candidate phyla radiation. These are sequences from organisms only known from their sequence, and we didn't know about these before this phylogram was created, that there's a whole group of organisms that have never been cultured, never been studied in the lab, that exist out there as a major group of organisms that we have not been aware of. And so this entire group over here needs now a great deal of study to find out what they are and what they're doing out there in the environment. Okay, I just want to talk a little bit about phylogenomics, and this is where now you take entire genomes, as in the last couple of phylograms I've shown, and find the common genes and then do a comparison. And normally the genes that are in common to both genomes that you can align, because often there are genes that are unique or can't be aligned because they're so variable. And those are concatenated. So you put one gene next to the next and the next and the next, and then you align those with the homologous genes from the other genome, and then you can make comparisons for many genes at the same time, a phylogenomic analysis. So initially, comparisons were made between 2 genomes or possibly 3, but that was quite a bit of computing time to do. But the computers and algorithms have gotten better, so now many genomes can be compared at the same time. In many cases, hundreds of genomes can be compared. And it's the same process where the genes are concatenated one to another, and then those are aligned with analogous genes from the other species, and then phylogenetic analyses are performed on those. So it would be great if all genomes were arranged in the same order like this, and that has to be done in the computer. But in real life, this doesn't happen. So although these are lined up in the center, if you look at genome A and genome B, these sequences come from very different places, and so they have to be found, located, concatenated in the same way, and then sequence alignment is done, and finally phylogenetic analysis is performed on the aligned sequences. So here's one example of an analysis that was done just to look at the synteny or alignment of the chromosomes in the grass family. So red is rice in the center. It has the smallest genome, and then going out to millet, sugarcane, sorghum, maize, wheat, and oats. And you can see in every case, some of the chromosomes line up very well. In other cases, there have been chromosomes that have fused or split. There are some chromosomes that are missing in some of these. So the chromosome numbers can change by quite a bit. The position of different sequences in the chromosome can be inverted or moved around to different chromosomes. So there's been chromosomal translocations throughout evolution. And so some chromosomes look very, very different, although the sequences on those chromosomes can be, in some cases, quite well conserved. So these are, you can see the arrows where they've split and arrows where pieces have been moved around onto other chromosomes in this alignment of the chromosomes in the grass family. So then when these chromosomes are compared, you have to assume that pieces have moved around. Sometimes there have been insertions or deletions of certain regions. Sometimes you get introns or transposons moving pieces around or interrupting certain genes. So these all have to be considered when realigning those sequences and genes. So here's just a theoretical diagram of some of this, where you have chromosomes splitting into several pieces, and then pieces being moved around, pieces fusing again in different places. And so overall, these have to be realigned in the final analysis for a phylogenomic analysis, but also modeling these chromosomes and looking at where the pieces are going. That's also a part of the phylogenomic analysis. In the case of bacteria, which many bacteria have only one chromosome, these same sorts of things would happen, but the chromosome would be a solid circle, but those genes still would be moving around on those circles from species to species to species. So one part of phylogenomics is looking at what genes and what gene families are in common and which ones are unique to specific species. And so 3 of these, Rhiza sativa, which is rice, maize, which is corn, and sorghum bicolor, which is sorghum, those are grasses. And then Arabidopsis thaliana here, which is being compared to it, is a dicot. The others are monocots. So about 55% of the gene content is in common with all of them. That's this one here. and then another 13.5% is in common with the grasses, so that's about 60-something%. And then you have some unique sequences in each one. So you can see a large proportion of the gene sequences, gene families are in common with all of these. And if you look further out with plants, you find the same thing. So almost all plants have a set of genes that are common to all plants, and that's about 50 to 60% of them. So that's about it for phylogenetics, phylogenetic networks, and phylogenomics. Here's a little bit about some of the software packages, and this isn't an exhaustive list, but just some of them. So this first one, NITSIS, and there are other similar packages. About $130, has UPGMA, neighbor joining, and others. Not a real useful system anymore, or a software package. Mega is actually used by quite a few people as UPGMA, neighbor joining, maximum parsimony, and I think the new one has Bayesian, I'm not sure. But that one's free, so if you want to try something out, that's probably the place to start. It's really fairly easy to use. Phylop here was primarily started to use maximum likelihood, so that's its strongest point. It has been altered, so it works. It can do maximum parsimony as well as some others, and it's also free. PALP is still available. It was written originally for parsimony, maximum parsimony, but now includes UPGMA, neighbor joining, and maximum likelihood. And I don't think it's been upgraded to do Bayesian yet. And then if you only want Bayesian, then Mr. Bayes, which is available online free, and there's more of an explanation. It's actually not too difficult to use, so that's another one to try out if you'd like. And here's just a simple evaluation of each. UPGMA is moderate to good in accuracy, fast and easy. Neighbor joining is also good to excellent, fast and easy. Maximum likelihood's slow, a little bit difficult to use, but it gives excellent results. Maximum parsimony also a little easier to use, and then Bayesian is a very good and excellent programmed set to use, and it's a little bit more difficult to use, but about the same as maximum parsimony. So those are just some of my notes on essentially how easy or difficult it was for me to use some of those. Okay, so that's the end of this video, talking partly about some of the other phylogenetic and phylogenomic and phylogenetic networks, and some applications that I've used in my own research. So thanks for listening, and I will talk to you later.